Welcome back, everyone. This is part B of the lectures on the electron transport chain. This series of slides will walk through the physiology and the steps involved in electron transport. Remember, the electrons from NADH and FADH2 have a lot of potential energy. The movement of the electrons through the chain produces the energy needed to pump hydrogen protons across its concentration gradient from the matrix to the inner membrane space. Now we are going to look at the electron transport molecules in the membrane that facilitate the movement of electrons and which create the channels for hydrogen protons to move across the membrane. First off, I want you to get familiar with the anatomy. I want you to see where the matrix is on the bottom side and the intermembrane space, which is on the north side of this picture. Then I want you to see that the Roman numerals indicate the fixed protein channels. These are enzyme complexes. Some of them are large and some of them are smaller. And we see that there are 4 fixed enzyme complexes. That means they're static proteins. That means that they're not moving. They stay embedded in the membrane. Okay, so these 4 fixed enzyme complexes are number 1, number 2, number 3, and number 4, all depicted in Roman numerals. Then you should see 2 mobile carriers of electrons. These are dynamic proteins. That means that they move. They're dynamic enzymes, and they're denoted as Q, which we're going to talk about, and cytochrome C, and both of these are circled in yellow. I want you to see also that at the end of the electron transport, we see the last electron acceptor is oxygen, which forms water. So we have a half of an oxygen molecule combining with 2 hydrogen protons to form water. Okay? Essentially, the whole process of electron movement through the complexes is to generate free energy to pump hydrogen protons across their concentration gradient to the intermembrane space. The fancy description for this is the cell needs to create a proton electrochemical gradient. This is a similar picture showing the electron transport system. I want you to highlight that there are 4 protein complexes. They are enzyme complexes, 1, 2, 3, and 4, depicted by Roman numerals. And then we have the 2 mobile carriers, Q and cytochrome C, which are not static. These are dynamic proteins. Q and cytochrome C can move, and they allow to help transport the electrons from complex to complex. Now let's discuss the electron protein complexes. Remember, the ETC involves a series of electron carriers that we just saw are arranged in order of decreasing free energy. They pull the electrons down the chain and ultimately transfer them to oxygen to produce water. And in the process, they release the free energy that's able to pump the hydrogen protons across the concentration gradient and create a proton gradient. There are 4 classes of carriers which we will briefly consider. These are grouped into 4 complexes called respiratory complexes. The first complex in the chain will accept the electrons directly from NADH. It is known as NADH dehydrogenase. Complex 1 will then donate electrons to complex 2, which will donate to complex 3, which will donate to complex 4. If the electrons are coming from FADH, they skip complex 1 and donate their electrons to complex 2. Listed on the slide are the names of the 4 protein complexes that are fixed in the membrane. The first one, NADH dehydrogenase. The second one, we've seen this enzyme in the citric acid cycle, known as succinate dehydrogenase. It accepts electrons directly from FADH2 in complex 1. The third complex is cytochrome BC1, which accepts electrons directly from complex 2. And then cytochrome oxidase, which accepts electrons directly from complex 3. So essentially, we're moving from a high free energy down to lower free energy, and we're transporting these electrons from complex 1 to complex 4, where then it will bind to oxygen to form water. There are also 2 mobile carriers in the electron transport chain. One is ubiquitone, also known as coenzyme Q10, and it receives electrons from complex 1, complex 2, and it carries them to complex 3. It acts as a little taxi to transport the electrons in the gap in the membrane. It also carries 4 electrons in total from complex 2 to complex 3. The second mobile carrier is known as cytochrome C, and this carries 2 electrons from complex 3 to complex 4. And these carry electrons between the protein complexes. So there's gaps between the fixed protein complexes, and these little mobile carriers act as the taxi to pick up the electrons and transport them between the complexes. One thing to note is ubiquitone is denoted as a big Q. You may have seen this before. Coenzyme Q10 is just a big Q, and so that's indicated in the figures that you saw earlier as the Q. Okay, just to make note of that. Some individuals may supplement with coenzyme Q10 in an attempt to try to increase the amount of ubiquitone, this mobile carrier, within the electron transport chain. The evidence to say that ubiquitone and the supplementation of coenzyme Q10 is affected is a bit up for debate, but there is a little bit of evidence to suggest it does increase. But a lot of it is perceptual because it's very hard to measure changes within mitochondrial concentrations. This slide provides a bit more detail on the arrangement of the complexes in the inner mitochondrial membrane. Remember, the electrons move from high free energy to a lower free energy at complex 4. The complexes live in the inner mitochondrial membrane. However, only complex 1, 3, and 4 actually span the entire membrane, crossing from one side to the other. And these are the only 3 complexes that allow for the transportation of the hydrogen protons across the membrane. Remember that the inside of the inner mitochondrial membrane will face the mitochondrial matrix, whereas the outside will face the inner membrane space, not the cytoplasm. I want you to follow the transfer of electrons down the chain and see that Q, ubiquitone, and cytochrome C are not fixed in the membrane. Rather, electrons from complex 1 and 2 are carried by ubiquitone to complex 3. And then cytochrome C is the taxi for the electrons from complex 3 to complex 4. Remember back to our discussions on competitive inhibitors? I mentioned that cyanide poisoning targets cytochrome C and inhibits its action, which stops the electron transport chain and causes death. I want you to get acquainted with this figure. Essentially, just pause the recording right now and look at the figure and try to understand it. Complex 1 oxidizes NADH to NAD. So this allows for 4 electrons to be passed from complex 1 to ubiquitone. And as such, it's a 1-to-1 ratio. So for every 1 electron that's passed, 1 hydrogen proton is pumped. So in complex 1, we see 4 hydrogen protons pumped across the membrane. Complex 2 oxidizes FADH2 to FAD. The combined electrons from complex 1 and 2 are carried by ubiquitone to complex 3, and 4 hydrogen protons are pumped across the membrane at complex 3. Cytochrome C then transports the electrons from complex 3 to complex 4, and 2 hydrogen protons are pumped across the membrane. So in total, we had 4 hydrogen protons on complex 1, 4 pumped across at complex 3, and 2 pumped across at complex 4, for a total of 10 hydrogen protons pumped across the membrane. Okay? The flow of the electrons releases the free energy for which hydrogen protons can be pumped across the inner mitochondrial membrane and against its concentration gradient to the inner mitochondrial space. This creates the electrochemical gradient. Now we have a higher amount of protons in the intermembrane space compared to that in the mitochondrial matrix. This is called a proton gradient. Before we end part B of the lecture series on the electron transport chain, I want you to watch this video again to get a visual representation of the electron transport chain. With the information you just learned, you will probably be able to understand the mechanics of what's happening a little bit more.